Topic for my today's presentation is schizophrenia. This presentation, we will first study about the neuroanatomy, neurochemistry, neuroimaging, genetics, environmental factors in schizophrenia, information in schizophrenia, neurodevelopmental models, and so the neuroanatomy. The following are the symptom domains of schizophrenia: positive symptoms, negative symptoms, affective symptoms, aggressive and cognitive symptoms. So the affective symptoms are predominantly from the ventromedial prefrontal cortex, cognitive symptoms from the dorsomedial prefrontal cortex, aggressive symptoms are orbitofrontal cortex and amygdala. The positive symptoms are from the mesolimbic circuit and negative symptoms are from the mesocortical circuit. So the dopamine pathways. So in brain, there are basically four dopamine pathways. The mesolimbic pathway, which runs from the ventral tegmentum to the ventral striatum. Then the mesocortical pathway, which runs from the ventral tegmentum to the prefrontal cortex. Then the nigrostriatal pathway from the substantia nigra to dorsal striatum, and the tubulo-interfundibular pathway from hypothalamus to pituitary. Out of which a mesolimbic pathway and mesocortical pathway have a role in schizophrenia. So normally in our brain, there is a balance in the dopamine level in the mesolimbic pathways and in the mesocortical pathways. So in patients with schizophrenia, there is dopamine hyperfunction in the mesolimbic pathways and dopamine hyperfunctions in the mesocortical pathways, leading to an imbalance, and hence the positive symptoms of schizophrenia because of the mesolimbic increased dopamine in the mesolimbic pathways, and the negative symptoms of schizophrenia because of decreased dopamine in the mesocortical pathways. Second is a glutamate hypothesis. So basically, we have all these glutamate circuits in our brain: cortico brainstem, corticostriatal, hippocampus-striatal, thalamocortical, corticothalamic, corticocortical direct, and corticocortical indirect. So in this diagram, as you can see, this is a they are pyramidal cells, which are glutamate-rich pyramidal cells. Then this is a cortical circuit, which we are depicting. So this is a glutamate pyramidal cell. Glutamate is an excitatory neurotransmitter. This is a GABAergic interneuron. When from the pyramidal cells, there is release of glutamate. It acts on the GABAergic interneurons through NMDA and AMPA receptors. So when it acts on the GABAergic interneuron, there is release of GABA, which is an inhibitory transmitter, and hence a negative feedback to the glutamate pyramidal cells. So in schizophrenia, as per the hypothesis, there is hypofunctioning of these NMDA receptors. So due to hypofunctioning, when the glutamate is released, it does not act on the NMDA receptors because of hypofunctioning. There is no release of the inhibitory neurotransmitter GABA, because of which there is no negative feedback. And therefore, there is increase in level of glutamate, and glutamate then hence via its extensions to the ventral tegmental area, there will be excess of glutamate and hence excess of dopamine. So dopamine hyperactivity is a downstream consequence of glutamate dysregulation in prefrontal cortex. Then third is our serotonin hyperfunction hypothesis, which is mainly applicable in psychosis related to dementia. So in dementia, it is considered that there is due to plaques, due to or the amyloid protein, there is a degeneration, or there is degeneration of the GABAergic interneuron, because of which there is imbalance between the inhibitory GABAergic and the stimulatory glutaminergic pathways, leading to serotonin hyperfunction and hence leading to dopamine hyperfunction. Then is GABA. There is loss of GABAergic neurons in hippocampus. So the GABA is synthesized from glutamate by glutamic acid decarboxylase, and we have two isoforms, GAD65 and GAD67. So in schizophrenic patients, there is reduction in GAD67 mRNA levels in the prefrontal cortex, and loss of this inhibitory GABAergic neurons could lead to hyperactivity of the dopaminergic neurons. Acetylcholine. The cholinergic neurotransmission is known to be integral to the cognition and memory function disrupted in schizophrenia. So decreased level of nicotinic and muscarinic receptors are reported in hippocampus, frontal cortex, thalamus, and striatum in schizophrenia. Abnormalities in the expression of central nervous system nicotinic receptors alpha 7 and alpha 4 beta 2 in schizophrenia. So post-mortem finding, there is decrease in M1 receptors seen in DLPFC and hippocampus. Now, neuropathology and structural changes. So there is these findings are seen in the postmortem. There is decrease in cortical thickness and total brain weight. Schizophrenia is associated with reduction in dendritic spines and total dendritic length. There is deficit in synaptic markers in the cortical pyramidal neurons. And these changes, they lack diagnostic specificity for schizophrenia. So in structural MRI and CT, we can see a reduced whole brain volume, reduced prefrontal and medial temporal gray matter volume. There is reduced thalamic volume, predominantly in the medial dorsal nucleus of thalamus. There is enlargement of lateral and the third ventricle and diminished total white matter volume. There is reduction in the hippocampal or amygdala hippocampal volume by 4%, which is lateralized to the left side. The basal ganglia, there is a volumetric increase in the caudate putamen complex. On electron microscopic studies, it suggests that there is increase in the number of synapses in the caudate and putamen, suggesting increased neuronal activity in the basal ganglia. Increase in the dopamine receptors, D2 receptor in the caudate and the putamen and the nucleus accumbens. Most of these morphological changes have been documented in the first episode of the psychotic illness and may be progressive. In functional imaging, we can do when we did a regional cerebral blood flow. During rest, there was no alteration in the blood flow. During activity, during active auditory hallucination, the blood flow increased in the ventral striatum, medial temporal lobe, and superior and middle temporal gyrus. During working memory task, the blood flow reduced in the frontal and the DLPFC activation. During problem solving and other executive functioning tasks, deficits in the DLPFC activation was seen. In PET scan, there was increased glucose utilization in temporal cortex during auditory hallucinations. Magnetic resonance spectroscopy showed prefrontal deficits. There was increased membrane phospholipid degradation and impaired synthesis have both been reported in the frontal lobe, and increased level of frontal lobe glutamine and glutamate. On a diffusion tensor imaging, white matter disorganization in the frontal and temporal subcortical zones. So the genetics, the heritability of schizophrenia is 75%, and it is the second most common heritable psychiatric disorder. So this data is derived from population epidemiological studies, family studies, twin studies, adoption studies, linkage studies, and chromosomal aberrations. So the population epidemiological studies, they say that the prevalence in the general population is 1%. In a non-twin sibling, it is 8%. One parent having schizophrenia, it is 12%, and both parents having schizophrenia, it is 40%. Family study is basically pedigree analysis. The data show the familial basis of schizophrenia with increased risk of developing schizophrenia in relatives of schizophrenic patients. In twin studies, the concordance rate was more in monozygotic twins, that is 50%, than in dizygotic twins. The linkage studies has limited role in schizophrenia. In genome-wide association studies, these studies help us to identify the chromosome locus of candidate gene in schizophrenia. These genes were identified. Dysynaptobravin binding protein 1, neuroglin 1, disrupted in schizophrenia gene, catecholamine O transferase gene, regulator of G protein signaling 4, proline dehydrogenase, D-amino acid oxidase, and D-amino acid oxidase activator. Most of these genes are involved in either myelination, neurogenesis, or in the synthesis of neurotransmitters. Now, environment and schizophrenia. It is said that genetic factors present from birth may interact with environmental factors across the life course to increase the risk of schizophrenia. So in this diagram, these dashed lines, they indicate the potential complex interrelationships between the identified environmental risk factors. Neurodevelopmental model of schizophrenia. So what it says is that the genetic predisposition plus the environmental, social, and psychological factors, they work together to form a neurodevelopmental abnormalities and target features, which cause brain dysfunction, in improper balance of the chemicals, and ultimately schizophrenia. So normal cortical development involves proliferation, migration of cells, circuit formation, that is dendritic arborization and myelination. The first two processes, that is the proliferation and migration, occur during the prenatal life, and the later two continue through the first two postnatal decades. The genetic predisposition with environmental insults lead to altered excitatory inhibitory balance in the prefrontal cortex and reduce myelination that would alter the connectivity. Inflammation and schizophrenia. So the activity of microglial cells is linked to the pathology of schizophrenia. So perinatal insults or early life stress prime this microglia to act in a hyper-responsive manner to later stress and encourage a shift to a pro-inflammatory M1 phenotype. So basically what they have said is the microglia exist in two phenotypes, M1 and M2. M1 is pro-inflammatory and M2 is anti-inflammatory. So when there is perinatal insult or early life stress, so the microglia are primed and they get shifted to a pro-inflammatory M1 subtype. Now, if the patient is genetically susceptible to schizophrenia, and also there is late life stress or immune activation, which will act on the prime microglia, leading to overactivation and aberrant synaptic pruning. So this is a diagram in which we can see in perinatal period there is a quiescent microglia, and if there is any stress in childhood, this microglia get primed. And if there is any stress in childhood or in adolescence, this gets get activated, and along with the genetic factor, there is inflammation, dysregulation, pruning, leading to this symptoms of schizophrenia. This is a two-hit hypothesis which may lead to excessive pruning of neurons by macroglia in stress-sensitive areas such as prefrontal cortex and hippocampus. Loss of synapse due to pruning by microglia lead to structural brain changes associated with schizophrenia and development of negative and cognitive. Now, the endophenotypes in schizophrenia. Endophenotypes are heritable and highly penetrant gene, also present in unaffected family members. They manifest as trait, that is, features that persist independently of the presence or absence of disease, co-segregate with the illness within families, and occur at a higher rate among non-infected family members than that of the general population. So a number of behavioral and physical studies have shown that early processing of sensory input is impaired in schizophrenia. Consistent with the behavioral findings, event-related potentials, that is ERP, show abnormality in early processing of sensory input. So P300 evoke potential. P300 is a large positive evoke potential wave that occurs about 300 milliseconds after a sensory stimulus is detected. So major source of P300 is the limbic system structure such as medial temporal lobes. So in patients with schizophrenia, this P300 is smaller. And in abnormalities in P300 wave, more is seen more commonly in children who are at high risk of schizophrenia. Similarly, there is sensory gating P50 deficit. Sensory gating refers to a centrally mediated inhibition of neuronal response to a no longer needed sensory input. So sensory gating is studied in laboratory using a EEG. The scalp EE register a small positive change in the electrical potential at 50 milliseconds following presentation of the stimulus, that is P50. If the same stimulus is repeated, the P50 is attenuated, and this is reduced in schizophrenia. And the last is smooth pursuit eye movement abnormalities. Human use a smooth pursuit eye system to maintain an image of a small moving object of interest within the fovea. Patients with schizophrenia are not able to accurately follow a moving target with their eyes. Similar abnormality in the non-ill relatives of schizophrenia programs are. These are my references. Sir, these are basically traits which are present in the in the relatives of patients with schizophrenia, who may, like they who may or may not have a disease. These are in early. the early detections?
Primary prevention. That is therapy which is we start giving antipsychotics to those people who are not yet psychotic. These antipsychotics is 50% rate, 50%, 100% so whether we should start giving antipsychotics to all one we start giving primary prevention. Primary prevention. Rights are good. That way we can test the primary prevention. Preventing the disease. Is it type of biomarker? It's been told us and it's supposed to be as well as the food stages to part of the same genetic condition. So biomarker it may not be genetically related, maybe lab finding, it may be a phenotypic presentation or maybe there is something genetic finding. So and the everybody genes, not all individuals express that gene. Even if that is present, it's not that actually explains why there is not present for every always between the monogram. So we have to take the DNA is the expression is there. Very important but there are practice and very important in this purpose. Neurobiology schizophrenia, physiology of schizophrenia, again, endocrine schizophrenia, dopamine aerobicis, dopamine aerobicis. Last time. This is very important because this kind of experience from psychiatry. Till now, it has been considered as psychiatry and medical. It is not of the main. They are but we are not able to develop enough investigation ability to diagnose and relate them very clearly. Hello. Structural findings that they are not very consistent. Hello. All the parts of the presentation are very clear और आप लोग को सब कुछ आना चाहिए इसका कुछ नहीं आएगा तो that can otherwise
So in our cortex, we have pyramidal cells. Okay, so these pyramidal cells, they have this neurotransmitter glutamate. And the glutamate neurotransmitter, these are the glutamate circuit which from cortex to brainstem, cortex to striatum, hippocampus to striatum, thalamocortical, corticothalamic, and there are corticocortical glutamic circuits. So these pyramidal cells, when they release glutamate, it act on the GABA interneurons. So these GABA interneurons, they release GABA, which is an inhibitory neurotransmitter, which will go and again act on these pyramidal cells, making it a negative feedback loop. Then And this glutamate, this act via NMDA receptors or AMPA receptors. So this glutamate hypothesis is NMDA hypofunction hypothesis, in which they say that this NMDA receptor is not working properly, because of which the glutamate will not act on this GABA interneuron. GABA will not be released. So this negative feedback loop will not work. If this negative feedback loop will not work, there will be continuous release of glutamate, because of which it will act on this cortico striatal pathway, making a increasing glutamate, which will ultimately lead to increasing dopamine and hence psychotic positive symptoms of schizophrenia.
So glutamate hypothesis is eventually related to dopamine. You must listen dopamine hypothesis to get understand glutamate. So and in next set she has mentioned that dopamine hypothesis, glutamate hypothesis is precursor of dopamine hypothesis. So down the stream, dopamine hypothesis is supposed to give it glutamate. Right?
Yes.
Receptors are working with against glutamate. डायग्राम पाथवे ग्लूटामेट इसमें डायग्राम में दो पाथवे दिख रहे हैं ना, एक तो ग्लूटामेट गाबा वाला पाथवे, एक ग्लूटामेट डोपामिन का पाथवे. ग्लूटामेट इज प्रॉब्लम डोपामिन. They are saying main problem NMDA receptor. NMDA receptor के हाइपोफंक्शन से ग्लूटामेट का फीडबैक बंद हो जाएगा तो ग्लूटामेट ज्यादा हो जाएगा, उससे वो डोपामिन ज्यादा कर देगा. डिफरेंट पाथ. Why different path? And the feedback, negative feedback is not. So otherwise there is no production of this thing, so it is different from the GABA.
The acetylcholine, basically they say that there is a problem in the acetylcholine receptors. So acetylcholine is an integral part for our cognition, memory function, which is basically disrupted in schizophrenia. So there is decreased level of these receptors, or there is abnormality in these receptors, because of which the dopamine does not act on these receptors, and which lead to the problems in cognition and memory. It is not related directly to the dopamine part.
Basically, this is one of the definition of cognitive symptoms of schizophrenia. And that's why it is a well-known fact that high anticholinergic prescription may lead to even further improvement of schizophrenia. That is why the first generation antipsychotics are supposed to have been known to cause, although they use the two process symptoms, still doing two process symptoms, but they are known to cause deterioration of cognitive. That is the next time treatment and still. To smoking, smoking in that is also like some there's only in the cognitive, but also auditory business. Is smoking based that is like there is some smoking. This part, people very far some way is also important. Patients may have it. So what is streaming KPN? That is also very important. What is streaming KPN? चार तो बना पॉजिटिव यूज द पॉजिटिव वे एंड नेगेटिव वे उसको इवैल्यूएट करता है वो पॉजिटिव का नेगेटिव भी जाता है.
So there is N30 this positive, so this positive wave, which is bigger, which is normal. In schizophrenia, this the smaller positive wave. This is like in normal people, we can when a sensory stimulus is given, so like we have this.
देखिए न्यूरोलॉजी में होती है टेंशन को टेंशन करते हैं
We have this ability to
Tension
filter.
Huh?
We have the ability to filter this sensory stimulus. But in
नर्व में सिंगल नर्व का नर्व में क्या होता है पोटेंशियल क्या होता है आपका सेंसरी स्टिमुलस लो पोटेंशियल मेलेबिलिटी का स्टिमुलस इज लो पोटेंशियल वो जो देखो कोई ट्रैवल ही करेगा ना उससे लो पोटेंशियल वो स्किज़ोफ्रेनिया वालों में कम होता है स्टैटिस्टिकली स्मॉल okay but in an individual you can't as a group you can differentiate that there but in an individual it is not like you can say ये schizophrenia so these phenotypes as a group we can differentiate but they are not significant specific enough to in an individual you can say कि इससे कम हो जाएगा तो ये schizophrenia का phenotype है that is why these are not that like effective अगर ऐसे कोई टेस्ट आ जाए हमारे पास में यहाँ पे स्पेसिफिकली हम बोलेंगे कि अगर इतने सेकंड से कम होगा चांसेस ऑफ साइकोसिस आर मोर और इससे ज्यादा हो गया तो देन वी कैन रूल आउट साइकोसिस ऐसा नहीं कर सकते बट मोटा मोटा ये है कि नर्व्स में कुछ तो प्रॉब्लम है दैट इज लाइक न्यूरोलॉजिकल डिसऑर्डर स्किज़ोफ्रेनिया इज नॉट साइकोलॉजिकल डिसऑर्डर इट इज न्यूरोलॉजिकल
remote potential की short.
ओके हाँ ये P300 पे आ सकता है ये वो पोटेंशियल बिकॉज़ like psychiatry is basically a branch of neurology. You should know neurons, how they're working. So in future, this is where all the interventions are going to be. You're studying brain channel mechanics. Okay, if they are more specific, you should know how you are stimulating the brain, how which areas, which nerve, which things are there. So earlier on, we used to spend a lot of time in defense mechanism, psychology, and, trying to do these things. So, because that was what we were doing, now this is the thing that because in the next 10, 20, 30 years, so it will become with using artificial intelligence. We will be able to like हो सकता है कि वो हमारे जो 30, 30, 300 कुछ ऐसा मोन्टाज बनाते हैं उनका मिला करके कि ईजी से या इससे आप जो है पोटेंशियल से आप डायग्नोस्टिक कर सकते हो फेस वाला तो आ ही रहा है सबको वैसे you do 30 minutes of interview and they will like assess अभी वो इसमें दिखा रहा था इसमें ये था कि वो उसने जैसे बनाया था उसने रस के जो रस होता है ना हाउ उसकी मसल्स के ऊपर बनाया था दैट वाज नॉट साइंटिफिक अकॉर्डिंग टू कि जो वो भाव होते हैं अक्सर असर ये सब उसके अकॉर्डिंग इसने फेशियल मसल का प्ले दैट यू कैन रिकॉर्ड और वो उससे फिर एआई रीड करके वो बताया कितने परसेंट टाइम तक आपका कौन सा वो था हाँ मसल तो उससे ये कि जैसे कि हम लोग देखते हैं कि भाई ड्यूरिंग द पेशेंट का अफेक्ट वाज रिस्ट्रिक्टेड सो यू हैव ऑब्जेक्टिव दिस थिंग क्या फेशियल मसल उसकी अभी
हमने वो पहले स्टडी की थी
हाँ
एक कंपनी दवाई थी
हाँ
वो जापान फंडेड स्टडी थी
हाँ
कि हम मतलब वॉइस सैंपल लेते थे और डिमेंशिया रिलेशन दिए थे तो उनका बोला था कि हम वॉइस की मतलब टोन से और वॉइस की बॉडी करके हम
Differentiate कर सकते
डिफरेंशिएट कर सकते हैं वो लेकिन वो जापान वाली कंपनी बैन करता हूँ तो बीच में
ओके
अब यहाँ पे वही
एक्चुअली
जो बायो फीडबैक वाला था ना, उसमें हमारे हमने वहाँ पे भेजी थी उसमें प्रॉब्लम क्या है कि बहुत टीचिंग बैकग्राउंड नॉइज के पीछे क्रैम्प था तो इसलिए मतलब वो भी हमारा सैंपल दे सकता है यहाँ का सब बोला इजी वाला तुमको इजी काम आएगा तो हम वही सामने वाले रूम में करे तो हो गया चलो 4-5 साल
ये बहुत प्रॉब्लम है कि स्माइल करोगे तो सेल्फी लेगा you can recognize smile and gesture करोगे तो gesture entertain कर रहा है easy और easy तो रीड कर ही रहा है easy भी जल्दी रीड करना जाएगा तो फेशियल एक्सप्रेशन रीड कर सकता है तो डू एन इंटरव्यू तो वो बता देगा तुम्हारा 50% of time you could difference or 20% time you can smile 8 times he was calling 12 times, तो उनको रिपोर्ट आ जाएगी साहब, MSc इजी हो जाएगी करना। और साथ में अगर स्किन कंडक्टेंस पल्स वगैरह, अभी तो देखो बहुत सी यू कैन, मतलब हार्ट रेट, ऑक्सीजन सैचुरेशन, स्किन कंडक्टेंस, यू कैन डू दिस। उस पर बहुत पढ़ रहा हूँ, उसको कैमरा के लगाओ, साथ में कर भी सकते हो।
तो इसलिए भी पता करें इसी तरीके से एंजाइटी और ऑफिस से कर एंजाइटी और इसलिए देखा उनका इतनी है
अच्छा ओके, हाँ
सपोर्टेबल है इतनी है इलेक्ट्रोड है और रिपोर्ट मतलब उसका व्यू मुझसे आपको होगा कोई ऐसा रिपोर्ट कभी जो
आपको मोस्ट ऑफ एफटी अप्रूव्ड है ना फॉर एरिथमियास, एरिथमियास लाइक यू कैन, एरिथमिया में वो कर सकता है, थोड़ा देख सकता है। चलो